Hi, my name is Abby Johnson. I'm a PhD student in the CMB program, and then I'm going to talk about my project identifying defined probiotics for use in commercial turkeys. So we want to find alternatives to antibiotics because stricter regulations on antibiotic use has caused a decrease in antibiotic use in turkey production. And so probiotics are beneficial bacteria, and they are a potential alternative that is very promising. And so what makes a good probiotic? So a probiotic needs to survive through the digestive tract, so the acidic environment of the proventriculus and gizzard, as well as the small intestine, which contains bile, and then they need to adhere well to host cells. And so I want to identify a strain or strains of bacteria that are specific to turkeys, which can be supplemented to them to improve their growth performance and prevent and treat infections. And so I'm focusing on Lactobacillus johnsonii. This particular species has been identified with high performance and low pathogen burden in chickens, and it is a natural resident of the turkey digestive tract, which is why I'm focusing on it. And for this, we collected 118 johnsonii isolates from turkey intestines over 10 weeks, and then we sequenced their DNA, and then we compared their relatedness and found that they were genetically different from each other even when taken from the same bird. They were more different based on geographic location, and then most importantly, when compared to different sources, especially the chicken, they were very different, meaning that there is potentially a host specificity. So I wanted to investigate how well my collection of isolates perform in different probiotic performance assays. So first I tested their ability to survive in bile using a bile salt hydrolase presence or absence assay, and I found that 51 of them were positive for bile salt hydrolase presence, and then 21 were negative, and then 14 had very low bile salt hydrolase presence, which is an enzyme that deconjugates bile. And then next I tested their ability to survive in an acidic environment. So again, in the proventriculus and gizzard, there's low pH of about 2.5. And so I measured their percent survival, so 100% would be the target. And then each bar is a different isolate. And as you can see, there is a lot of variability in the isolates in their percent survival. And so next I wanted to see if I could identify any genes that were correlated with probiotic performance. So I took all of the different genes from our entire collection of isolates, and I looked at the gene presence or absence within them. So first I looked at, so we identified 4,089 genes which were found in all the isolate, or in the entire collection of isolates, and then there were 1,341 that were shared within all 118 isolates, and then 970 that were shared between 113 and 17 isolates. Then interestingly, 1,778 isolates were only found in between 16 and 1 strains. So only 30% of the genes are shared between all the isolates, even though they are the same species. So I wanted to identify specific genes that were associated with high probiotic performance. So I input gene presence or absence, as well as, in this case, bile salt hydrolase activity, presence or absence. And I'm discussing bile salt hydrolase activity because that's what I have finished. And so we're able to correlate phenotypic presence or absence with gene presence or absence, and is able to predict potentially correlated genes. And I identified 441 which were statistically significant, and 334 of them unfortunately were hypothetical proteins or not annotated. But I was able to identify 2 previously identified bile salt hydrolase genes. So the next step, I want to test all 86 viable isolates on the acid tolerance assay. I've only tested 30 so far, and then I want to do the host cell adhesion assay, and then identify the potential genes correlated with both of those assays. And then we want to use the genes that I've identified with high probiotic performance to screen future candidates, as well as genetically engineer superior probiotic with high performing traits.